Hello, everyone. Very big good evening to all of you. Am I audible, everybody? Can I get some quick response in the chat box? Are you all able to hear me? Thank you so much, Carol, for your reply. Thank you. All right. So, welcome to the session, everyone. I hope all of you had a good festival yesterday and your preparation is also going on side by side. You people are not focusing on festivals that much now, and your main focus is on CSIR net. So, how's your preparation going on, everybody? Are you people practicing enough questions? Do you think this Brahmastra series is helping you? Everyone, whatever you think, please let me know. See, we'll be covering all the important topics from the syllabus in this Brahmastra series. But along with that, if you people especially want some topic to be covered, okay, you are facing any problem in solving questions from a certain topic and you want that topic to be discussed here, so please let us know in the comment section. Then we will definitely cover that and we can plan our classes and sessions accordingly. Yes, can I have some replies, please? See, guys, it is very important to communicate in the class, right? And this is a class only that is going on. And if you people are not communicating with me, how would I know that you people are there? So, as you all know, today we are going to discuss questions from the half unit of Unit 8, Inheritance Biology. So, from subunit F, that is Microbial Genetics. Pedigree analysis, since we have already taken in a different session, so we'll not cover pedigree analysis. We'll be for sure covering polygenic inheritance, which is a topic from where a lot of questions are coming in the past few sessions. That is what I have checked. Then we'll be covering little bit of mutations as well. As I told you in the last class that mutation is one of the very important topic from where a lot of questions come, and lot of permutation and combination can happen with mutation and with other topics in the unit. Okay, so that is what we are going to see today in today's class, some very typical difficult questions from these segments. All right. And I really hope that you all will enjoy the session. Please stay till the end of the session. You know, a freebie is coming your way. So there will be a document with more questions from the various varieties of this, from the subunits of unit 8F to 8K. And with the explanations, not only questions, but with their explanation also. So for that, please stay till the end of the session. Okay, so Carol, if it is going slow, then you have to pick up the speed, okay? Because only very few days are left, not like barely, it is 1 month that is left. So this is the time when you have to, this is like the, even half of the last lap of a marathon. So this is the time you have to pick up the speed, okay? So if you are lagging somewhere, so whatever concept that you don't know, just leave it. There is no point of learning anything new. Just revise whatever you have studied. Start revising now. Revise, solve questions, any problem, go back and revise again. Okay, that should be your strategy. Don't try to learn anything new now. Whatever you have learned, keep revising them now. All right, so let's get started, everyone. So, this is the first question. I definitely want all of you to try to solve it along with me. So, this is a question from polygenic inheritance, and here we are talking about the influence of environment and genotype on the genotype. So, given below are figures representing four different situations or examples of genetic and environment, that is, temperature effect on plant height in two varieties of a plant species. In figures A, C, and D, solid line and dash line represent the mean values of plant height for the two varieties G1 and G2 respectively. In figure B, solid and dash line overlap. Okay, so in figure B, you can see that there is only one line. So it is clearly mentioned that because solid and dash line, they both overlap here. Okay, now what is solid and dash line? So it is also mentioned here that so solid and dash line is for two different varieties. Okay, so G1 is shown by solid line, G2 is shown by dash line. Given below are four statements explaining the four figures, right? One, two, three, four. Which one of the following options correctly matches the figure and their corresponding explanation? Okay, so what you have to do is there are four statements here, there are four figures here. Okay, so you have to match these statements with the figures that are given. Yes, I'm giving you 2 minutes to solve this question, and please let me know your answer, everyone. Yes, everybody, please try to solve this question quickly. Can I have some answers if you have already solved it? All right, so let's have a look at how to solve this question. So, in this question, what we see here is this graph. Okay, now you will be able to answer this question if you have a good understanding of how to read the graph. Okay, now this how to read a graph, this is an analytical skill that you must have. If you don't have, then you develop it, because this will help you in solving part A, part B, as well as part C type of questions. Beautiful colors and Carol. No, I'm sorry. Thank you for trying, but I'm sorry, that is not correct. Yes. So what do we have here? Okay. So first graph. Okay. So now how do you read the graph? So whenever there's graph given, okay, not only for this question, for any other question, whenever there's graph given, always read the coordinates first. Okay. So on y-axis, we have plant height. On x-axis, we have temperature. Okay. So in this particular graph A, what do you see? That plant height is for G1 genotype, plant height is shown by this solid line. For G2 genotype, dashed line. And on x-axis, there is temperature. So is the temperature showing any effect in the plant height? Are the plant heights increasing for G1, G2 with the temperature? Is it decreasing with the temperature? The answer is no. So temperature is not causing, because this is the complete flat line that we are getting. So with increasing temperature that is there on the x-axis, there is no increase or decrease. So there is no change in the plant height. Okay. So what this graph will show? So this graph will show that this is in this particular plant, the plant height is caused only by genotype. Environment has no effect. Okay, so which statement is this? That is statement 1. Says that plant height is influenced primarily by genotype of the two varieties, and because with temperature there is no effect or no change in the plant height, so temperature or environment is not causing any change in plant height, or it is not influencing this particular tree. Okay, so statement 1 is for graph A. Right, now once we have analyzed this, so again, as I always say, that solve your question in part, so we can check the options, and from there you can eliminate option 4 and option 3, because A1 is not there. Okay, 1 and 2 is there. Right, now let's check for B. So in B we have only one graph. So this graph or this line is for G1 and G2 both. Okay, so what is it now? This graph, G1 and G2, they are different genotype, but still the graph is same, means they are producing the same plant height, but the height is changing here, because it is not a straight flat line here. There's an increase, there's a slope in the line, but this increase is according to what? It is increasing according to temperature. As temperature is increasing, height is increasing. Okay, so what we can say, that because G1, G2 both are producing and showing the same change in height, and it is showing the same height and same change in height, so genotype doesn't have much effect in this particular trait. So this trait is primarily getting governed by which factor? By temperature. Okay, so which statement is that? That will be statement number 2, that variation in plant height is influenced only by temperature and genotype has no effect. Because genotype has no effect, that's why G1 and G2, they both are showing the same graph. Okay, so with 2 it is B. Now with 2 it is B, automatically you can eliminate option 2 and you have got your answer. The same thing that I keep telling you, and I have told you many times, that when we solve question in parts, okay, and you check or you analyze the options like this, so you, most of the cases, you will not have to solve the question till the end. Okay, halfway also you are solving the question, you most probably you will get the correct answer. Okay, especially that is true for solve the this match the following kind of questions. All right, so in exam, wherever you have matched the following kind of question, try to attempt them first. They are the easiest in all questions that are given in the question paper. Okay, now for C, what do we, so let's analyze this graph C also. So in graph C, G1 and G2 has different height. Okay, means genotype is influencing the height. Okay, along with that, what we see, that temperature, with temperature, height is also increasing. Okay, means this particular height of the plant is not only getting governed or is not only influenced by genetic factor, but also by temperature. Okay, so genotype and temperature both are having an additive effect on plant height. So that when with genotype and also with temperature, as temperature is increasing, height is also increasing. Okay, so statement 3 is for graph C. And D, what do we here, so in here G1 and G2, two different genotypes, two different trait. Okay, two different heights are there. Okay, but here with temperature there is either increase, so there is an increase in height for G1, but there is a decrease in height for G2. Okay, so in this case in D, so how is D different from C? So in D also, genotype as well as temperature is showing an effect in the plant height, but the effect of how temperature is affecting G1 and G2 is different from each other. Okay, it is not affecting in the same way. Plant height is not increasing in both the cases. In one genotype, plant height is increasing, another one it is decreasing. So both genotype and environment have an effect on plant height, with the two varieties responding differently to the environment. So statement 4 is for graph D. So A1, B2, C3 and D4. Answer is option 1. Do you all follow, everybody? Am I clear, everyone? Okay, let's move on to another question. Now, this is a type of question again which has been asked multiple times in the CSIR paper. Okay, now this is a topic which is deletion mapping, and this deletion mapping map related question has come like 10 years back also, if you see it is there, 5 years back also, if you see it was there, and like in recent times, especially the post COVID time also, if you see, question has come. Okay, in fact, this question it came in June 2024 exam only. Okay, so this is, let's have a look at this question. So as always, I want you all to analyze it, give me some answer, and then we will continue. Okay, yes, so it's your time, everyone, to analyze this question. So this is a reminder also for all the students that if you have any confusion in deletion mapping, or if it is a topic that you have skipped, so please go back and revise it from unit 8. Deletion mapping is something that you should not skip. It is that important. Yes, if you have solved it, I would definitely like to know what answer are you reaching to. Options are given here, 1, 2, 3, 4. Which one do you think is correct? Okay, so it's time for us to see this question. Very good, beautiful colors. Yes, your answer is absolutely correct. Okay, so mutants or bacteriophages that carry deletions can be used to rapidly locate mutational sites of newly obtained mutants. The mapping is based on whether wild type recombinants can be recovered when the deletion mutant and the novel mutants are brought together. Four independent deletions, 1 to 4, of a region were used to map four novel mutations A to D. Deletions starting from a fixed site are shown below, and the lines denote the region to be that has been deleted. Okay. Now the results of mapping are summarized in the table, where plus denotes recovery of wild type recombinant, minus inability to do so. Further, it was observed that out of four novel mutants, no revertant was observed for mutant A. Mutant B and C do not complement each other. Okay. Now, based on this, that first of all, based on this data that we have got after deletion map, the crossing that we have done, and also by keeping in mind these two statements, following conclusions are made: A, B, C, D. So you have to identify which of the combinations are of all correct statements. Okay. Now, let me give you a little background on deletion mapping. So, deletion mapping is a technique which is used for physical mapping of genes on the chromosome. Okay. Now, what is the idea of deletion mapping? Is here also we will do a cross. The cross is done between an individual who is heterozygous for deletion and who is homozygous and the other person is homozygous recessive for that particular trait. Okay. Now the F1, okay, so the next generation, now we will analyze the next generation. If the next generation is showing the wild type phenotype, that will be possible if the wild type allele is present. Okay. Now how can the wild type allele be present? So if a wild type allele is of that gene is present, means the gene was not deleted. Okay. So in F1, I will write it down here. So F1 result, right? So in F1 result, if we get a wild type, okay, what does it denote? That the dominant allele or the wild type allele is present. So if the wild type allele is present, means that particular gene was not present in the deleted segment. Okay. So if wild type wild type progeny we have got in the F1 generation, means gene not present in deleted segment. Is it making sense, everyone? If wild type phenotype we get in F1, that means gene is not present in deleted segment. Okay. If we get a mutant phenotype in the F1 generation, that means gene present in deleted segment. Okay. So if gene was basically present in deleted segment, means the gene has been deleted. Okay. Right. So now what we will do is, before we go in this table here, let's have a look at this series of deletion. So deletion 1, 2, 3, 4 are given. So before we go here on the table, so we will imagine that suppose we are talking about a particular gene that is present in this segment, okay, a segment which has been deleted in all four. Okay, so any gene that is present in this particular segment which is deleted in all four will show what progeny in the next generation? It will all show negative, right? Negative means gene was present in the deleted segment and it has got deleted, right? What about a gene that is present in this particular segment? Means a segment which is not deleted in deletion 1 but deleted in other 3. So for deletion 1 it will show a wild type phenotype, for others it will be negative. Okay. Similarly, a gene which is present in this particular segment, the segment which is present in deletion 1 and 2 but absent in 3 and 4, so it will show a phenotype of plus, plus, but minus, minus. Okay. And in this particular segment, if you have any gene, so it will show plus, plus, and plus, but minus for deletion 4. Okay. Now, once you have analyzed this, you just have to match it with the table that is given. Okay. So which is the this mutation which is showing negative for all the 4 deletions? So it is deletion. It is this mutant A. Okay, means mutant A will be present in this particular region. Am I clear, everyone? Can I have some response? Guys, are you people following? Please respond in the chat. Okay, continuing. All right. Uh, so continuing with this B. So B will be present in a region where it will be plus plus and minus minus. Okay. So where it is, that is this particular region. Okay. So plus plus and minus minus. Okay. Now, which is the mutation that is showing plus and minus minus minus? That is mutation D. That means D will be present here. And C is plus 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 minus. So C is present here. Okay. So this way what we have done, we have mapped the mutations on the chromosome. Okay. A, D, B, C is the order in which the genes are present. Okay. So the conclusions, if you check, it says A, mutation A lies within the region of deletion 1. Okay. Yes, mutation A lies within the region of deletion 1. So this statement is correct. B, mutations can be ordered in A, D, B, C. Yes, that is exactly the order that we have found out. A, D, B, C. Okay. Now see, earlier when question used to come from deletion mapping, it used to be only this much. Okay. Where you just have to find the order in which the genes are present or the mutations are present. Okay. But this time the question was even made more complex, as it was combined with another concept, that is complementation. Okay. So here you see these two lines. Okay, what was observed? That out of 4 novel mutants, no revertant was observed for mutation A. So this is linked with mutation, that there was no revertant we have got for mutant A, and it also says that mutant B and C do not complement each other. So if B and C do not complement each other, in the last class also, Herbert, thank you so much. I'm glad that you found it helpful. Please practice it. Okay. Yes. So coming to this question, so mutant B and C do not complement each other. So in the last class also we have talked about complementation test. I hope all of you understood that. So if B and C do not complement, means B and C are happening in the same gene or they are happening in the same cistron. Okay. So B and C are allelic in nature, right? So when the A mutation cannot be reverted, so what all mutation are cannot be reverted? So there is transversion and definitely there is deletion, like larger deletions, they can't be reverted. Okay. So if statement C says that mutant A could be a deletion, that is correct. Okay. Now, statement D says that mutants B and C are located on two independent cistrons. So, if B and C are present in two independent cistrons, then they would have complemented each other. But since they do not complement, means they are present in the same cistron. So, it is incorrect. Okay. So, what all are the correct statements? A, B, and C. And that gives your answer as option 1. Is it followed, everybody? Okay, before going ahead, this is our announcement for Lakshya batch. We have already started, but still you can still join our Lakshya batch. Last enrollments are still going on. So if you are preparing for June 2025 as well as for the December 2025, please join our Lakshya batch. You get CSIR net as well as GATE coaching. Along with that, you get molecular biology internship absolutely free, and this molecular biology internship is going to start very soon. Okay. And along with that, you have all these extra features that comes with Biotechnica. So please, everyone, if you need any further information about pricing or discount, so please contact this toll free number and you will get all your informations. If you are interested for the coaching, please join Biotechnica as soon as possible, because we have already started the classes and the courses, the topics are going on. Please join and don't miss out. All right, moving on. So let's see the third question. This is based on pattern observed in DH or F2 progeny identity. Which of the pattern A to C are based on DNA markers that are allelic? Okay. See, before that, what is mentioned here? The following gel patterns are that of DNA markers observed in parents P1 and P2. F1 from a cross between them and double haploid progeny panel A, or F2 progeny derived from selfing of F1 in panels B and C. Okay, so you have three panels A, B, and C. A double haploid is a genotype formed when plants were developed from haploid cells which have undergone chromosomal doubling. Okay, that is double haploid. Now, based on the pattern observed in DH or F2 progeny, identify which of the patterns A to C are based on DNA markers that are allelic. Okay. Now, again, in the last session also, I told you that from nowadays, questions are coming, like question from any topic from Unit 8 can come where it is linked with DNA markers. Okay, so markers is something which has to be very, very solid. This concept of markers have to be very, very solid with you all, and you should be very much well versed with reading the markers in a gel. Okay, right. So can I have some answer here, please? What do you think? Which of the panels out of A, B, and C are showing DNA markers that are allelic in nature? Yes. So I hope all of you have analyzed the question. Let's have a look at this question now. Okay, now what are double haploids? So it is mentioned that double haploids are produced from plants which are developed from haploid cells that have undergone chromosomal doubling. Okay, so suppose you have a plant which has this genotype. Okay, now at the time of gamete formation, the alleles will separate. Okay, now when the alleles have separated, now you have got the gametes which are actually your haploid cells. Okay, now when they undergo doubling, chromosomal doubling, that is when you will get the double haploid. Okay, so if chromosomal doubling of this cell happen, so it will become capital A, capital A. If this cell happens, it becomes small a, small a. Okay, now you can take any genotype, you can consider multiple genes also, but when the alleles are getting separated in the gametes, the haploid cells, so when the double haploids are generated from there, double haploids will always be homozygous. Okay, so this is something that you should keep in mind, that double haploids will always be homozygous. They can never be heterozygous. Homozygous always. Okay, now in plant 1, I'm sorry, Herbert, that's not correct. So in panel 1, what we have, so there is P1, P2. Okay, so P1 has this band, P2 has this band, F1 has both the bands. Okay, and in the double haploid which is produced from F1, it has this band also and this band also. Okay, which is actually showing that, see, a P1 and P2 has this band and F1 has both the band. Okay, so this is basically showing a marker which is, first of all, it is codominant in nature because heterozygote is showing both. Okay, and when we create the double haploids, so double haploids are having is homozygous for this one or the other one. Okay, so since here either this one or that one is present, that means these are the allelic markers. That is, they are present at the same location, but these like they are alleles of each other. Okay, so A is allelic. It is showing allelic DNA marker, right? Because the homozygotes have only one of them. Okay, so A is allelic. What about B? Now in B we do not have double haploids. Instead we have the F2 progeny. Okay, now and in F2 progeny also, like in this particular case in B, so F1 has both. Now F2 has all the three varieties. Okay, it has only this marker, it has only this marker, and it has this heterozygote also. Okay, now this again will be the scenario when these two markers are alleles of each other. Like if this individual is capital A, and if this is small a, F1 will be capital A small a. Okay, and then when F1 cross happens, then we get all the three varieties, that is capital A, capital A small a, and small a small a. Right? So panel B is also showing allelic marker. Okay. What about panel C? Now, panel C here, okay, so there is a very minute hint which is given, and that hint is here. Okay. So in this particular group, is it like nothing was placed here? No, this is F2 progeny. Okay, sample was put, but none of the band appeared here. Okay, now if none of the band happens for an allelic marker, then basically that particular individual do not have the allele, means the particular individual do not have the gene itself. Okay, and will it be able to survive? No. But if this F2 progeny, if this group of F2 progeny is surviving even without any allele here, okay, that means the marker that we have considered here, these two bands are not from alleles of the gene. Okay, they will be different genes. Okay, so panel C is not allelic, only A and B are allelic. Okay, so answer will be option 4. All right, so I hope all of you followed this. So guys, again a reminder that DNA marker, this concept have to be as absolutely clear with you all. Next question. Now this question was also given in last session exam only, and again this question, like for answering this question, again you just have to develop the skill of reading a graph. Okay, so I'm giving you a couple of minutes to analyze this question. Okay, let's have a look here. So this is the name of two scientists. They carried out an extensive mutation screen to identify all genes involved in segmentation of Drosophila. The graph shows results of analyzing mutations on chromosome 2. Okay, so this is the graph. Now I told you, whenever you have graph, the first thing is to identify the coordinates and understand the correlation. Okay, so in on this coordinate you have number of segmentation genes. So that is being shown with the solid line. On x-axis, we have number of progeny analyzed, which is 1,000, 2,000, 3,000, 4,000 till 5,000. And this dotted line, what it is showing? It is showing the number of segmentation mutants. Okay, so solid line is for number of segmentation genes. Dotted line is for number of segmentation mutants, okay, which is coming here on this scale, and the number of progeny analyzed. Okay, now based on the above figure, which one of the following option is a correct statement? I'm sorry, beautiful colors. Thank you for trying, but your answer is not correct. Okay, so let's have a look. Statement 1. Okay, what it is saying that analyzing 10,000 progeny would have led to identification of substantially more number of segmentation gene. Okay, so we are stopping the analysis at 5,000 progeny. It is saying that if we would have continued our analysis till 10,000 progeny, then the number of segmentation genes might have increased more substantially. Okay. Now, see, is it the trend that this segmentation gene is showing? This solid line is for segmentation gene. Okay. So the number of segmentation genes identified was increasing till 2000 progeny. Okay. Once we, after this particular point, from 2000 to 5000, you can see that there is barely any increase in the number of segmentation gene. Okay, so when there is a barely increase in number of segmentation genes, so if instead of 5000, if we would have gone till 10,000 also, only few more segmentation genes only we might have discovered. That is what the trend is showing. If we extrapolate it, still it would be very flat line. Okay, so it will not increase the number of segmentation gene. That is incorrect. At least that is what our graph is showing. That is what you have to analyze. Okay, what is the trend of the graph, and if we extrapolate, what will be the result? Statement 2 is saying that analysis of progeny beyond 2000 led to identification of new alleles of already identified genes rather than more new genes. Okay. Now, after 2000, what is happening is that this dotted line is increasing, right? It is increasing with the same slope. So, why is it increasing? Now, what is this dotted line? This is the number of segmentation mutants. Okay, so even beyond 2000, number of segmentation mutants are increasing, but the number of segmentation genes have not increased that much. Okay, so all these mutants, where they are having the mutations, so ideally there can be only one explanation, that all these mutants are having mutation in one or the other of this already identified genes. Okay, so if two or more mutations, if they are happening in the same gene, what do we call them? Allelic mutations, because they are the one which will be producing the alleles of the gene. Okay, so beyond 2000, analysis of the progeny led to identification of new alleles of already identified genes rather than new genes, because new genes number is not increasing, but number of mutants are increasing. So where these mutants are having mutation, they must be having mutation in the already identified genes, right? So it resulted in the in the identification of new alleles of the already discovered genes. So this seemed correct. What about 3 and 4? Let's have a look there also. More dominant mutations would have been observed in the first 1000 progeny. Now we don't know about dominant or recessive mutation. Okay, nothing is described here, and just by looking at the graph also, we cannot say whether the mutation was dominant or recessive. No information is given regarding it. Okay, fourth curve would remain the same irrespective of the mutagen used. That also is incorrect, because this curve will vary with the mutagen that we will be using, and because that will depend, like with the mutagen changing, the process, the chemical process of how it is causing the mutation will also change. Okay, so the curve would it will not remain same with the mutagen. Okay, when we change the mutagen, the graph and its uh, overall nature of the graph will change. Okay, so answer will be option 2. Only statement 2 seems like the most correct answer. Moving on to next question, since we are running out of time, so let's see, quickly move on to the next question. I hope all of you are able to follow the questions. Yeah. So the plaque morphology of wild type and R2 mutants of T4 bacteriophage following infection of different E. coli strains is summarized below. The following two experiments were carried out. Experiment 1 and experiment 2. Okay. Now, what was experiment 1? Co-infection of two independent R2 mutants on E. coli strain resulted in several plaques, all being small and ragged. Experiment 2. E. coli B strain was infected with above R2 mutants. T4 phages from the resulting plaques were used to infect E. coli K strain. Few plaques were obtained, which were all small and ragged. Based on the above observation, the following statements were made. A, B, and C. Which of the following option is the combination of all correct statements? Okay. Now, what is given here is that for this T2 phage, T4 phage, wild type is small and ragged colony. Okay, in B strain also, in K strain also. R2 mutants, they cannot grow in K strain. They can grow in B strain, but the type of colony or type of plaque that they form is large and round. Okay. Now, what was experiment one? So I'll quickly just draw it in a scientific manner, not just in words. Okay. So we had E. coli strain. Okay. And in this E. coli strain, we infected it with two independent R2 mutants. Okay, let's say R2 plus R2 dash. Okay, these are two different mutants. So they both infected the E. coli cell, right? Resulted in small plaques. So this infection resulted in small plaques, and those plaques were small and ragged. Okay. Now, small and ragged. Which kind of T4 phages formed small and ragged plaques? The wild type. Means here we are getting the wild type cells. Even though we have infected R2 mutants, two independent R2 mutants, but when they infected the E. coli strain, so they produced wild type progeny. Small and ragged plaques means it is wild type. Okay. What is experiment 2? In experiment 2, E. coli B strain was infected with the above R2 mutants. Okay, so here we took E. coli B strain. Okay, and again this B strain was infected with R2 and R2 dash, these two mutants. Okay, T4 phages from the resulting plaques were used to infect E. coli K strain. Okay, R2 and R2 dash, they both infected here. Whatever plaques were formed, okay, they were used to infect E. coli K strain. Okay, R2 and R2 dash anyways cannot grow in K strain. Okay, so the plaques that were formed, that was used to grow in K strain, and it was found that they all formed again small and ragged plaques. Okay, fine. So now let's have a look at the statement. So statement A says that experiment 1 indicates two mutants are allelic. See, this particular experiment where you are combining, where you are allowing E. coli cell to get infected with two different R2 mutants, and you are checking for the progeny. So in the progeny, if you find wild type variety, means this is actually complementation that is happening. So R2 and R2 dash, when they are growing in E. coli cell, when they both infected E. coli cell, so inside the E. coli strain, they complemented each other. Okay, and they formed the wild type variation, and that resulted in the formation of small ragged plaques. Okay, so when they can complement each other, when R2 and R2 and R2 dash are having mutations that are happening in different cistrons. Okay, so if they are happening in different cistrons, will we call them allelic? No, it will be non-allelic. Okay, so experiment 1 indicates two mutants are allelic. This is incorrect. A is wrong. Okay, now A being wrong, automatically you can eliminate option 1 and 3. Let's have a look at option B. The statement B. Experiment 2 indicates that the wild type T4 phages that infected E. coli K strain resulted from a recombination event. Okay, now this experiment 2 actually it shows recombination. Okay, that we allowed both the strains to grow in E. coli B, then they will recombine there. Okay, they will recombine and result in the formation of wild variety. This wild variety, when it is infecting the K strain, it will produce small and ragged plaques. Okay, so this actually shows recombination event. So statement B is correct. So I have got this image also. See, this is the image that you will find in many of the books also. So when R2 is this, and this is R2 mutant 1, this is R2 mutant 2. Okay, so when they both are infecting the B cell first, Okay, so that is how they will recombine. Okay, so when they are recombining, they will produce two varieties. One is A plus B plus, one is A minus B minus. So A minus B minus will not be able to grow, but A plus B plus will be the wild variety, it will grow. So when we take this plaques and infect them to the K cell, then we will get the plaques. Okay, so who all are forming plaques? This recombinants are forming the plaques. Okay, so this shows recombination. C statement says that in experiment 2, if T4 phage isolated from B strain are used to infect the E. coli B strain again, all plaques will be large and round. Okay, see, large and round will be the plaques that are formed by this R2 mutants in the B strain. Okay, but as I told you here, that this recombinants will also grow. This one, it cannot grow. Okay, what about this A plus B plus? It will grow, and it is wild type. So if it is wild type, it will produce what kind of colony? Small and ragged. Okay, so when we infect it again in B strain, so it won't be only large and round. There will be large and round, and in fact, large and round will be the parental type. So they will be more in number. But at the same time, small and ragged plaques will also be there, which will be showing the recombinants. Okay, so this statement is also incorrect. Which is correct? Only option B. This is the correct answer. Okay, now I took this question because in last two sessions, like December also, in December 2023 also, there was a question from this. Okay, a similar question. And we saw a similar question in June 24 also. Okay, so this is something that if you have not practiced, if you have skipped, so please go back, pick up the reference book and have a look at this concept. Okay, again guys, you will be able to answer it, you will be able to understand it if your understanding of complementation test is sound. Okay, now this is a question that I have from bacterial genetics. Okay, microbial genetics. 4 different HFR strains of E. coli were mated with F minus recipients, and the time of entry of various donor markers were found to be as below. Okay, HFR1, HFR2, 3, and 4. Based upon the above observation, the following statements were made, assuming MET to be at 0 minute and 3O to be at 15 minute. Which one of the following option represent all correct statements? Okay, now this, for this particular question guys, I will give you for your homework. Okay, I want you to try, I am giving you hint. Okay, so when this kind of question is there, so this is based on interrupted mating. So in case of interrupted mating, what is the concept? That we will interrupt the conjugation at different time intervals. The genes will pass through the F minus from HFR to F minus with in the same order how they are present in the bacterial chromosome. Okay, so the time lapse between the transfer of two adjacent gene will help us to find out the distance between them. Okay, that is why you see here the distance between the genes will be given in minutes. Okay, now even this this type of question is there, what you have to do is first you make a circle. Okay, because bacterial chromosome is a circular DNA, and then you start placing the markers. Okay, so for HFR1, let's say it is MET. Okay, and then there is 3O. So if I put MET here, let's put 3O here, and the distance between them is 15 minutes, right? 30 minus 15 is 15. Okay, then 3O and Phi. So I'll write Phi here. Distance is 42 minus 30. So it becomes 12. Okay, Phi and Mal. Okay, if you minus the time, 57 minus 42 is 15. Okay, so likewise you have to map all the genes on this bacterial chromosome, and then you check this statement. Okay, that starting with MET. So if MET is getting transferred at 0 minute, then is histidine getting is located at 59 minute? Then if azai is at 74 minute, if cysteine is at 11 minute, if mal is at 46 minute. Okay, so you have to check these statements. So, and that you will be able to check only when you have placed all these markers, okay, HFR 1, 2, 3, 4. Fine. So when you check, when you do that and you check for it, you will get that the answer will be like these statements 1, 2, 3 are correct. Okay, 4 is incorrect. Okay, so guys, please you try to do this question on your own. All right, because we are running out of time, and I will show you not this question also, this one. Okay, now this is a very interesting question here, and it is little difficult also. So transduction was used to map the FAD-L gene, the result of two-factor crosses between FAD-L and two linked markers, PER-F and arrow-C are shown below. Which one of the following is the correct map of these three genes? Okay, so you have this donor, you have the recipient, and you have the recombinants. Okay, numbers obtained are given here. Now, when we have to make the map, so first of all, it is transduction. So we have to look for co-transduction frequency. Okay. Now, for co-transduction frequency, what is the understanding? That the genes which are located close to each other, they will have higher co-transduction frequency. The genes which are present far away from each other, they will have lower co-transduction frequency. Okay. So co-transduction frequency is inversely proportional to gene distance. Okay. Now see, donor is FADL and PER-F. Now recipient will be the opposite, and then we have selected PER-F plus. Okay. So the cells which have got the second marker as FADL, that is 200. The cells which have got second marker as FADL plus is 800. Okay. So if you have to calculate the co-transduction frequency between FADL and PER-F, which number you should count? Okay, that is the major decision that you will have to take. Okay, so I'll just write it here. So I'm finding the co-transduction frequency. Okay, so co-transduction frequency of FAD-L and PER-F. Okay, now see, in the donor, okay, whatever genes are present together, if they transfer together, okay, that is when we say co-transduction. So PER-F plus in the donor is present with FADL. Okay. So which class of cell you will consider? You will consider the cell which has got PER-F plus and FAD-L, not FAD-L plus. Okay. So this number will give you the co-transductant. So these are the number of cells where this two in the same combination how it is present in donor has passed. Okay, so co-transduction frequency will be how much? Number of cells which has received both the genes from the donor divided by total number of progeny. So total number of progeny, when you add it all up here. Okay, so for this, you can see this is 1000, right? So we are considering one gene here. Okay, so this is 1000. So 200 divided by 1000, that gives you 0.2. Okay. Next, in the same way, find the co-transduction frequency of other two. So FAD-L and arrow-C. Okay. Now again, we are checking for arrow-C plus. So which category we have to consider? We have to consider the one with FAD-L because our donor has FAD-L. Okay. So this is the class we will consider. So that will be 400 divided by 1000. So 0.4. Okay. Similarly, co-transduction frequency of arrow-C and PER-F. Okay. So again, we are checking for arrow-C plus. So we have to look at this PER-F, this combination. This is 500. So this will become 500 divided by 1000. So 0.5. Okay. So now what do we see? That co-transduction frequency is inversely proportional to gene distance. So the two genes which has the least co-transduction frequency will be having the maximum distance between them. So FADL and PER-F has the least co-transduction frequency. So if I make a gene map, then FADL and PER-F will be present in opposite sides. So if I place FAD-L here, so at the opposite end, I will put PER-F. Okay. Right. And the co-transduction frequency between them is 0.2. If you convert it into percentage, it will become How much? 20%. Okay. Now, what about ROC? ROC definitely will come somewhere in between. So, ROC will come in between where FADL to ROC is 0.4 and per F to ROC is 0.5. Okay, it is almost in the middle, but still we see that ROC's co-transduction frequency with per F is little higher. So, ROC will be little towards per F. Okay, so ROC will be little towards per F. Then only ROC and per F's co-transduction frequency will be higher, which is 50%, whereas FADL and ROC's co-transduction frequency will be 40%. Okay, right? So now if you see the options that are given, the maps which are given here, so you can eliminate option A and option C, okay, because FADL and PERF are not present at the end, okay. And when you check for B and D, so here as you see that the co-transaction frequency between FADL and ROC is 40%, not 60%, okay. So that's why this is eliminated. Answer is option B. All right. So thank you, everyone. So we come to the end of this session. Thank you, everyone, for joining. And here again, announcement for the Lakshya batch. If you are interested, if you know somebody who is interested, please let them know the toll-free number of Biotechnica, and you can call and ask for more information. And you can download today's free document of more questions along with explanation given here in the chat box. Please go ahead, download it, practice those questions, see whether you are able to solve them on your own or not. If not, take help of the explanations and try to solve other questions also. So keep attending this free sessions, everyone. Every day we are having this session from 6 to 7, and from Monday to Friday. Please join these sessions. They are completely free. If you know somebody who is preparing, let them know also about this free sessions done by Biotechnica. Thank you, everyone, for joining. See you again in next session. Have a great evening. Keep practicing and keep preparing for your exam. All the best, everyone.